thank you all for coming today. This is our first citizen science lecture for the school year. My name is Ramya. I'm the citizen science coordinator for the Biscayne Community Foundation. If you don't already know, we have, starting at the beginning of the school year in September, we have a citizen science lecture every third Thursday of the month. Today we have with us Dr. Margaret Miller from Secore International. She is their research director and studies corals and coral restoration. And so she's going to talk to you about the projects that they're working on. Before I begin, I just want to say I'm really glad to see so many students here today. If you're here for the key challenge, make sure you sign in on the sign-in sheet in the back so that I know that you're here and I can tell your teachers. If you have a cell phone, please put it to silent before we get started. And that's it. So thank you all for coming.
Thank you. Thank you all for coming. Hopefully you'll get to learn a bit about corals and coral reef restoration tonight. I'm sure here in a place like Key Biscayne, folks are a little bit more savvy about some of these topics than a lot of places, but I thought I'd start with some basics. When I go talk at actual schools and to kids, I usually start out telling them that corals are a lot like Legos, right? We call them modular organisms because they're made of little bits. They're made of little bits just like Legos where you have a little brick. Their biology in that sense is very different than an organism like us, right? When there's a baby human and that baby human grows, it doesn't grow more arms and legs and heads and things like that, right? It's just all of it gets bigger. But when a modular organism grows, it essentially replicates that unit. And so in the coral, that little unit we call a polyp. And that polyp essentially has a mouth and a bunch of arms and a digestive tract and a reproductive system. And so as the coral grows, it adds those little units, just like you would add when you're building a building out of Legos. So here's another kind of funny thing about corals. The classic sort of question, right, about animal, vegetable, or mineral. We usually think of those as separate categories, right? So which one would you call? Let's have a show of hands. Who would put corals in the animal category? All right, that's good. How about the vegetable category? Anyone? Anyone for vegetable? Anyone for mineral? Okay, I got a couple for mineral, right? Because that's true. So what I'm going to say is yes, all of those, right? That's one of the things that makes corals really cool. They are animals, so you are all correct there. They're also all plants in a sense because they have these little tiny cells that live inside them that are little plant cells that live inside their tissues. But up in their tentacles, in that tissue, is where those little tiny plant cells live. And so corals are able to derive their food, most of their food and energy from the sun, the same way as if they were a plant. And so this is a pretty amazing symbiosis that enables them to garner enough energy that they can actually create rock at the same time. So the skeleton of the coral, so in this picture, this dark part down here, the stripy part, the bottom part of the coral is the skeleton. That skeleton is actually what makes limestone. And so all of the ground that we live on here, anywhere you're in a limestone cave, those are all the type of rock that's created by corals as well. So animal, vegetable, mineral, yes, all three. So that's pretty neat. And then in addition to that, just kind of like we're learning that humans and human health is really dependent on the microbes that we have living inside us, right? Our gut microbiome, all those bacteria and things that live inside our guts. Corals have actually a whole nother component of microbes also that live as part of their organism, similar to us, except that corals, all their microbes are living on the outside. They're covered with a mucus layer, and so that mucus layer on their, more or less like their skin, is where that microbiome resides primarily in coral. Some of it's inside the tissue as well, but that outer layer is really important. And we're actually just really, on the research side, beginning to barely understand that this microbiome is really important to the health of corals as well. So that's a whole nother complicated part of the biology that we're just really beginning to learn about. These special symbioses and the fact that they can create rock, these characteristics mean that corals provide a lot of different values, both for ecosystems, but a lot of values that human societies benefit from. And you all are familiar with a lot of these. How many of you all go fishing offshore here? right? A lot of the fisheries around here are reef fisheries, so the corals build structure, again, architecture, that are, a lot of the reef fishes use as habitat. How many of you are divers? Do you enjoy diving in the area? So again, the beauty and the diversity that corals create makes them attractive to divers and tourists. And so there's a lot of economic value derived from coral reefs. But another really important one that we're beginning to understand more and more, you can kind of see the indication. This is depicting how coral reefs, again, because they build that wall, create a barrier along our shoreline. It's like a wall that protects our shorelines where coral reefs are intact. And as we are experiencing more and stronger hurricanes and sea level rise and all of these assaults onto the shoreline, coral reefs, the value that coral reefs provide in terms of protecting that shoreline are going to be more and more valuable into the future. And so that means we need to be more concerned. You can kind of see from this picture, I think, see where all the waves are breaking out here? Where you see waves breaking, that means that the energy from those waves is being dissipated. And so those waves are not able to cause as much damage and erosion to the shoreline. So this is an example where a bunch of smart people with a lot of numbers sort of calculated the force in those waves that are coming against the shore. And in fact, in areas, as I said, where you have this intact reef crest, up to 90% of the wave energy is attenuated by that reef crest. And so that's energy that's not, you know, causing that house up there on the beach to get washed under, or causing erosion on the beaches and this sort of thing. So this is a really important value that coral reefs provide. And the corals are the one, by building that rock, the ones that create that structure. Probably a lot of you are also fairly aware that corals are kind of in bad shape, right? They're not doing well. Many corals, especially in our region, especially in Florida, but throughout the whole Caribbean region, have undergone tremendous declines in their populations. And we're talking 90, 95, 98% less coral for certain species than we used to have. So there's a lot of reasons for this. There's a lot of different threats and factors that are causing coral decline. Here in Florida particularly, disease is one of the things that we observe as sort of the ultimate cause of death. So especially over the past few years, there's been a tremendous amount of coral in South Florida that's died from disease. This is partly probably a result of thermal stress. Again, with global warming, there's a lot more heat stress on these animals during most summers, especially here during 2014 and 2015. Those two summers, we had extremely warm, sort of an ocean heat waves, and corals did what we call bleaching. This is what you see in the picture up here. This is was a relatively healthy stand of Elkhorn coral that bleached completely, and all of that coral died in the summer of 2014. So just a couple years ago. This picture was taken at Grecian Rocks, which is one of the popular snorkeling sites just off of Key Largo. So these repeated thermal stress events cause a huge amount of mortality. And then we have a host of local stressors as well, like pollution, the water quality problems, physical disturbances when we do have storms or hurricanes. There's also natural local disturbances like predation, right? There's things that eat coral as well, which normally wouldn't be a problem, but when you have that added on to all these other things, it really causes mass coral mortality events. And so that's a problem. The partner problem that goes with that is that most of these Caribbean reef-building coral species are simultaneously showing recruitment failure. So that means we don't have baby corals that are joining the populations to replace the ones that have died. And so together, when you have too many corals dying and not enough new ones joining the population, that ends you up as an endangered species, believe it or not. And this is something also that's sort of brand new. I mean, just in this decade, we have 7 species now of our local coral species that are listed under the Endangered Species Act. The first 2 were listed, the Elkhorn and Staghorn coral were listed in 2006. And then we had 5 other species that were listed in, I guess, 2012, I think it was. One of the characteristics about these listed species that I think is worth noting Generally, we expect endangered species, right, to be the rare species, species that are rare to begin with, right? And so because there aren't very many of them to begin with, they're more likely to be endangered. But in fact, 5 of these 7 species that are endangered corals used to be the dominant species, right? They were the ones that made up most of the corals on the reef and that built most of that reef structure. They were the abundant and dominant species, and those are the ones that we have listed because the population's declines have been so great. So that's not a very good situation. And this is kind of an illustration of what that kind of looks like here in the Florida Keys. This is a photo series that was taken by a gentleman named Gene Shin. He worked for the U.S. Geological Service, did a lot of work on coral reefs in the Florida Keys. Back, you can see this first photo is from 1961 up in the corner. You can see that big, healthy coral. Moving through the decades here, you can see this beautiful Elkhorn and Staghorn coral that are very healthy. Between 1971 and 1988, you can tell all that Elkhorn and Staghorn coral is gone. It's all dead, all right? And then since then, you see this kind of what's happened is what we have now at this same site is really dominance by these sea fans and these soft corals. So it's been a very noticeable, I think visually noticeable transition where we don't have those hard corals building that structure, building that habitat for fishes or that protection for our coastlines within that community anymore. And this is typical of many reefs throughout the Caribbean. And so as we've sort of recognized how bad the loss of corals is, there's definitely been an expansion in interest and application of different types of coral restoration activities. Really, as recently as about 10 years ago, here in Florida, and I think in most sort of reef management community, there was the approach that was generally used was there is reef restoration that we'll think about, but it's mostly in these cases of like an acute human disturbance, such as something like a ship grounding, right? This is actually a ship that grounded in the Florida Keys back in the mid-90s. And there was a project to sort of rebuild some of this reef structure that was damaged with boulders and things like that. So this is kind of the activity that was implemented as reef restoration for quite a long time, up until, as I said, fairly recently. But with this expanding sort of threats from heat stress and the mortality that we were talking about, we really are coming to understand that just removing the stressors, that those coral populations are in such bad shape that they're really not able to recover on their own. The natural population growth that those species should be capable of has really been impaired. As an example, the frequency now of these major thermal stress events, this is a depiction of this sort of the global extent of these thermal bleaching events in our modern times. The frequency of these events is down to about 6 years, all right? Maybe in the past they occurred maybe every 20 years or 30 years, and now they're down to a frequency of every 6 years. And that just means that there's not enough time for corals to recover in between that very frequent disturbance. And so what we're really involved with very actively now is actually coral population enhancement. So we're really propagating corals and working to get new corals growing on the reef. So that's kind of what we're going to be talking about the rest of the talk. There are sort of 2 main ways that you can propagate corals. That corresponds to the 2 main reproductive modes that corals use naturally. So if you're a branching coral, most branching corals are able to propagate from fragments. So that means if a branch breaks off, it's able to reattach to the reef and keep growing and make a clone of itself, essentially. That creates clones. And this is very common for branching species of coral, and this is a benefit for certain types of coral propagation. Many of you may be familiar, I'm sure, with the Rescue a Reef program here locally. Have any of you aware of that or participated maybe in some of their, they have some citizen science programs here locally where the corals, as I said, they're fragmented. Elkhorn and Staghorn coral are particularly amenable to this approach. They're fragmented. They're able to grow fairly quickly on these tree structures they use in offshore nurseries and create a lot of coral that they can then transplant back out onto a reef to restore a population on the reef. Now, the other means that we can use to propagate corals is a little bit more difficult, but it means starting with baby corals, all right? This is what a baby coral looks like. Would you guys recognize that? Well, that's what we're gonna learn a lot more about baby corals tonight. So this is the other approach. As I said, it's a little bit more difficult, it's a little bit more complicated, but the benefit is that when we create baby corals, we get an increase in the genetic diversity of the population that ends up. When we're fragmenting, as I mentioned, that creates clones. So that means we can create a lot of coral, but it's only going to be the same genetic individual that we started out with. All right, so there's limited genetic diversity when we use that approach. When we're using baby corals, we're going to get an increase in genetic diversity because we're mixing moms and dads the same way that that we understand, and that is the way that we get greater genetic diversity in subsequent generations. And so this is the approach that Secore is really focused on, and we'll be talking a lot more about that. The baby corals are important, as I mentioned, because sexual reproduction is the means that populations get greater genetic diversity. And the reason this is important, this is just a sort of a little depiction. We know that the environment is changing really fast in the ocean. There are temperature changes, there's chemical changes that are taking place in the environment. And so our best understanding, based on conservation biology, is that genetic diversity and large population sizes are the best tools that populations can have for adapting to these types of environmental changes. And so the idea is we want as many different colors of balls in our population so that we have a few that are going to be able to survive and thrive in future environmental conditions. And so this is the reason that we are invested and working really hard to perfect these methods that we can develop means to propagate from baby corals. So this is gives you a little bit of an idea about a coral life cycle. It's kind of complicated. And essentially, all of these stages that I kind of have in the circle, these are all life stages that are really hard to observe. Like, you can't go out into the ocean really very well and count how many coral larvae there are in the ocean. Just doesn't work that way. A lot of these stages, they happen in the middle of the night, and they're microscopic little dots in the ocean and this sort of thing. So one of the things to think about is that we don't really know what the normal or natural success or process, or to know whether we're doing well in terms of the settlement rates that we get. We don't have the natural number to compare it against, because these are largely processes that we can't observe. So most of what we understand about all of these different steps comes from laboratory studies that have been done. So this is kind of a different view of those different stages, but that you can kind of see a little bit more realistically what they look like. So again, the species that we work with are called broadcast spawners. So the eggs and sperm are both produced by the same colony, so they're hermaphroditic. The eggs and sperm are packaged together, and they're spawned, these little bundles that you see here, what you see in this picture, those little dots, are little bundles of egg and sperm packaged together. They're released by the parent colony just a couple of nights, generally, per year. So these spawning events are rare. We have to figure out when they're going to happen, and then we have to go out there to the reef, as I said, mostly in the middle of the night, usually when there's a thunderstorm or other stuff going on, to try to be there to actually collect the spawn as it's being released from the parent colonies. Then we need to mix that spawn from different colonies together. Even though they're hermaphroditic and each colony produces both eggs and sperm, they have to cross with a different colony in order to have successful fertilization. So that means it's a term we use is self-incompatible. You have to have a mixture of different parental genotypes in order to get fertilization. But you can see then cell division starts, and over the course of a week or so, the embryo goes through this process of development, and then at a certain point, it starts swimming. At a certain point, it develops behavior, and it'll swim down towards the bottom. It will seek out a surface somewhere where it undergoes metamorphosis. So similar to what a caterpillar turns into a butterfly, right? This very radical change in its body, it attaches itself onto the bottom and it turns into a polyp. And so this is that polyp that we saw on the adult colony. That's the first one. And then within a few weeks it starts building skeleton and then eventually can become a whole new big coral again, although that takes quite a while. They grow relatively slowly. So this is kind of how we start our assistive processes. As I mentioned, when the corals are spawning in the middle of the night generally, we have to sort of anticipate when that's going to happen and be out there, and we use these sort of tent structures that we can place over the coral and collect those bundles as they're being released from the parent colony. I put this photo up here too just to emphasize that these coral nurseries can actually be a really good source too for spawning stock. So they have lots of coral of known parents, like we know which individuals are which, and so that can be a really good source where there's a lot of coral in one place that we can efficiently collect eggs and sperm as well. And then there's all these different steps of fertilization and carrying those larvae through all of their developmental phases. That Secore has developed over the years processes to help with. And we work a lot with both professionals from zoos and aquaria, professional husbandry experts, and scientists as well in developing these methods. And so we've been successful at being able to assist in each of these steps at a small scale. So we can reliably predict when the corals are going to spawn, more or less. We can collect a lot of eggs and sperm. We can get them to fertilize. We can get them to settle onto these artificial substrates that we use. But we can do that for maybe, a few hundred to a few thousand per year, all right? There's enough coral restoration that needs to happen that we really need to get that to many hundreds of thousands or even millions per year. And so that's really the emphasis of our work right now. As I said, they start out so small. This is just to give you an idea, all right? This is a centimeter in a little tiny dish. Can you see these dots even? They're virtually microscopic. And so that's the size of the larvae that those corals start out, all right? They can end up, a quarter the size of this room as adults. Yeah, do you have a quick question?
Is the purple thing coral?
So the purple thing here is a little piece of reef rubble. This has a crustose algae on it, is what's pink, and that's a good habitat for baby corals to settle on. Yeah, so that's not a coral. That's just a little piece of substrate that I put in there to give them something to make they think that they would want to settle. But yeah, they're so small. And so even after they settle, that means that what's around them tends to be a jungle. Even though what grows on the bottom of the reef is, we would not notice it, but when you're the size of a baby coral, it ends up being a jungle. And so that ends up being one of the main hurdles that baby corals have to face after they settle, is that competition from those plants. And the circles there you can see. This one's really hard to see, but here you can kind of see the mouth, and then this is the little skeleton of the first polyp of a baby Elkhorn coral. Can you see it? Just barely?
I just realized what it was.
So they have a tough beginning to life, that's for sure. So as I said, we've been successful in raising baby corals then up to adulthood in small numbers. So this is an example. Our team in Curacao has been working for quite a few years, and this is a baby, well, a not so baby Elkhorn coral. Now this is an adolescent Elkhorn coral here that's 4 years old, and has, at the age of 4, has begun spawning itself in synchrony with the natural population that it came from. And so this is sort of our success looks like that, which is an Elkhorn coral that's joined the reef and has begun reproducing itself in concert with the populations on the reef. So Elkhorn is a relatively fast-growing coral. Some of these slower-growing corals, Colpophilia natans, is one of the brain corals. And you can see when it's 3.5 years old, all right, not quite 4, but it's only about this big. All right, that Elkhorn coral up there at 4 years is about this big. So there's a lot of differences between species also in terms of how quickly they're able to kind of grow up and join the reef, so to speak. But these brain corals, even though they grow slowly in the beginning, they're the massive brain corals, Copophilia natans. And so they can create big structures on the reef. So as I said, we've had success with small numbers of these baby corals, but we're really interested in getting more of them and getting more of them to survive on the reef. And so there's two factors that we've been working on to try to improve efficiency, essentially, our efficiency in producing these. One is developing substrates like this. You can see, and I have some here. I'm going to pass some of these around in a little bit. This is like the one that you see in the photo. These are concrete little structures. They're made with a mold. They have little grooves in them. But these are designed, I don't know if you've ever seen like a big riprap like embankment, right? So they're the structure is modeled after those, where they're kind of a self-stabilizing shape, such that a lot of the time and work that it takes, for example, to outplant those fragments of coral is the time it takes to glue them down. And that can be true for substrates for baby corals as well. But the idea of these is we're not going to glue them. We're just going to find a little nook or crevice in the reef and just pop them right in. And because these are a self-stabilizing shape, they're able to be retained pretty well. And so the point of that part of the shape is to make it a lot faster and less work to outplant them onto the reef. Then the other aspect that we're really working on is just raising more larvae at a time. When we raise them in the lab and we have to keep them in buckets or big tanks or things like that and change their water a lot, it limits the number of larvae that we can raise. So we're really looking to develop these very much larger pools, essentially, that we keep in the ocean so that the larvae are able also to be closer to their natural environment, and we can raise a lot of larvae at a time. And so we can put these structures, these little substrates, directly into the pools. The larvae go through their entire life cycle in that pool, settle directly on here, and then we can take these directly and put them onto the reef. So that's the idea in terms of being able to improve the efficiency and then also the survivorship of those babies after they go out to the reef. So this, I have a teeny bit of data here, you'll have to forgive me, but I'll just let you know that this is sort of based from a pilot study, again, that we did in Curacao, with a little golf ball coral. Actually, this isn't the big reef building corals. But essentially, when we use them that way, we settled a bunch of larvae onto these substrates and put them out on the reef. When we settle sort of 20 to 30 babies on each of these substrates, after a year, we have about 70% of them that still have a live coral on them. So we think that that's a pretty good ratio of success in terms of getting one-year-old corals out on the reef that we can do quite efficiently. The other aspect that we're thinking about a lot, and it's going to vary both by, I think, the environment, but perhaps the species as well, is how much investment it makes sense to grow these corals in a protected environment before we put them out on the reef. I think a common concept where you have a different amount of investment in an individual offspring. Humans have pretty high investment. Things like frogs or fish and stuff, they have a really low investment in each offspring. And there's probably an optimum for different types of environments and different species. And so that's partly what we're still learning for baby corals. There's different approaches. So this is what the approach I'm just describing for you here in the middle. But here you can see some baby Elkhorn corals actually that the folks at Mote Marine Lab grew out for a year in the lab. So they were about this big before they were placed out on the reef, instead of being those little microscopic polyps. And so they've actually done extremely well. You can see they started on these little plugs and they've grown out across the reef, and those are doing quite well. But that was a big investment to grow them in the lab for a whole year. On the other extreme, these are some folks working in the Indo-Pacific and the Philippines. They took the larvae directly and settled them back in this big tent that they put directly on the reef. So they're bypassing this substrate step that we're using by just taking the baby corals and settling them directly on a reef. Now again, that subjects them to all that jungle and this sort of thing that I was talking about before. In some regards, or some places or some species, this may be successful, and then that would be more efficient than using the substrates. But this is where this is an area of research to sort of figure out for different places, different reef conditions, what will work. So this is just also to emphasize that, you know, part of what makes coral reefs excellent is the diversity, right? If we restore just Elkhorn and Staghorn coral to a reef, that probably isn't restoring the community and all the functions that we want on that reef, because we really want to address a lot of different species when we're doing reef restoration. And so this is just a few of the species on Caribbean reefs. Secore is working right now actively with about 6 or 7 of these species that we're actively collecting and culturing larvae and settling them and placing them back out on the reef. Part of the goal then is to use these different species. They have different characteristics, and some of them are better suited to less than perfect environments. The Elkhorn and Staghorn coral actually are very sensitive species, and those are the ones that we've kind of started working with the most since they're endangered. But they really do need very healthy environments. Whereas some of the brain corals, like I was talking about, they may be able to occupy a much wider range of habitats. And so they may be more successful if we use them for restoration in reef areas that aren't quite pristine, where there's more challenges for those species. So we want to be able to use many different species and be able to mix and match them a little bit to the environment that we're wanting to restore. And then this is another aspect that I want to sort of emphasize for the future. These are areas as we think towards the future where we're going to have an ocean that's much warmer. I mentioned this coral bleaching, that's a huge problem. And this is just to show you some examples where corals in natural populations, or in this case in the nursery population, you can see in this picture, these ones across the top row are all bleached, right? Those are different fragments from the same genotype. This U25 is the same parent colony that all of those were cloned from. You can see the middle one that's called K3 is bleached a little bit. Can you tell that? Like the tips look kind of white, but the main part of the branches still looks healthy. And then this bottom one looks perfectly healthy, even though it's been under the same temperature stress as the others. So we have this natural variation in the population that we're culturing, where some of these are able to persist much better under this situation of heat stress. This is a similar example of 2 colonies of star coral, again, living right next to each other in the same environment during this heat and bleaching event in 2014. You can see one of them is perfectly normal, and one of them is severely bleached, and this colony probably died, because usually if it's going to look that bad. But again, so if we're going to be raising corals and restoring corals, we'd like them to have this characteristic, right, as opposed to this characteristic. So we're beginning to think about wise ways that we may be able then to leverage this variability in the population to be able to increase the resistance of the populations that we're restoring, so that they're going to be better able to survive in our future environments. And so there are different types of processes that are being researched, essentially. This is really in the realm of the future. These are areas of research. They're not things that we're directly implementing right now in restoration, but they're important things to consider in the future. Things like, so by stress hardening, what I mean is, sort of giving the corals, maybe the juveniles or corals that we're going to be using for propagating, a little artificial heat stress, right? Give them a little bolster and see how they do. And the idea being, right, what doesn't kill you makes you stronger. So if there's ways to treat corals in ways that can make them more resilient or more resistant to heat stress. Selective breeding is an example where if we can choose parents that have those resistance characteristics, we may be able to produce offspring that are more resistant as well. And so those are aspects that I said. They're under research right now, because these approaches also contain risks, right? These are not approaches that we've used in conservation in the past. We haven't had to. But we're thinking about them very seriously for the future. So to kind of summarize, coral restoration, and especially through breeding and raising baby corals, these types of interventions can yield a large number of baby corals, right? When the corals spawn, they produce a large amount of spawn, and we can raise a large number of corals. So there's a big source of material that we can grow new corals with. We're really focused on upscaling, that is, these technologies to develop in situ rearing of larvae and different new types of substrates. We've developed some new prototypes. I showed you the older one, but these are ones that we've sort of dreamed up that are testing right now with our field teams throughout the Caribbean. I'll pass these around if you want to kind of see what they look like. They're made of ceramic. They're all 3D printed. You can pass a few of these around. So we have baby corals settling on these sort of right now. We're in the process of outplanting them so that we can evaluate the survivorship of the baby corals on these different shapes and different microhabitats. Hopefully they can make their way around. We'll make sure everybody gets to see them. So this is a major aspect of the research that Secore is undertaking, as I said, both to make them easy and quick to outplant, but also to give them that habitat, those nooks and crannies, or whatever that habitat is going to be, that's going to help them survive against that jungle, those competitors that we were talking about. But an important point we need to not forget when we're doing restoration is that the environmental quality still needs to be able to support corals in our coastal environment. So we need to improve management and climate change mitigation so that our coastal oceans can support corals at all. And we need to not lose sight of that when we're doing restoration. So there's things that everyone can do to help, right? Important ones are taking your own actions as best you can to mitigate the climate change risks. So that has to do with what you eat, how you use energy, and how you get around, and also how you vote. So those are all things that everyone can participate in curbing the effects of warming that are so devastating to corals. Minimizing single-use plastics. Again, it's less of a direct effect to corals probably here in this region, but in the Indo-Pacific, there are studies that show where there's a lot of plastic debris in the water, and that plastic often will snag on corals, and it's one of the main, well, not one of the main, but it's a very important vector of that coral disease effects, are from plastic bags and plastic debris. That's important. Sustainable seafood, be careful about the sunscreens that you're wearing when you're going in the ocean, or even not. Conserving water and being aware of the water that you use, what goes into your drain, and what goes onto your lawn, because all of those substances are eventually going to end up in the ocean, in our coastal ocean. And I'll recommend, there's a documentary, it's available on Netflix, called Chasing Coral. And I'll recommend that to you as well. It tells very well the story of this mass global bleaching event, coral bleaching event, that happened a couple of years ago. And it explains really well this connection of coral endangerment and climate change. So I want to acknowledge Secore's lead partners that support our work, both financially and through scientific collaboration. I am happy to have some discussion and questions as folks would like. Yes, a question.
What do corals do for us? Like, why do we need them?
Why do we need them? That's a good question. We talked about that a little bit. There are different reasons, there are different benefits that corals provide to us. A lot of it has to do with that structure, the fact that they make rock, right? So they build structure and they build reefs. And so those provide both habitat for a lot of other types of organisms that we like lobster. Do you like lobster? No, not you. A lot of people do. There you go. A lot of people like those. Lobster and fisheries of various sorts. But they also build this wall, as I said, that protects our coastline. So it protects from erosion and flooding and things like that. And that's the other main benefit that we get. Plus, they're just really beautiful, don't you think?
Yeah.
Yeah, and that's important too. Yes, in the back.
I heard somewhere that coral and algae makes like 75% of all the oxygen in the world.
Yeah, and I have to admit that corals don't make as much oxygen as the algae do. Exactly. So that is a common number I hear, that, yeah, all the oxygen, a lot of the oxygen that we breathe does come from the ocean. Most of that's made by planktonic types of algae, so those different kinds of little cells and stuff like that that are plants, but that live in the water column of the ocean.
Wait, I'm not done. Are you broadcasting this?
I don't think it's broadcast. I understand that it is going on YouTube later. There is a YouTube channel where it will be posted.
I'll just go on it later.
Okay. Yes, in the back.
But you also mentioned that part of the reason they're not able to bounce back is because of recruitment failure. So the baby corals aren't growing, they're not attaching. Do we know why they're failing to recruit more than in the past?
We know it's probably similar to the things that cause them to die. There's actually a bunch of different things. If you remember all those different stages, I think there's a lot of reasons. And again, these are not they're difficult to document the relative importance of these different things, as I said, because they're hard to study. But I think to some extent, the parent corals are so unhealthy that they're not producing eggs and sperm that are healthy. And so to some extent, the larvae aren't being produced in the first place. So the larval supply is less than it should be. The corals, these little microscopic blobs, but they have amazing sensory capacities. And this is something that research has really shown. They can detect sounds. They respond to sounds that are characteristic of reefs. They respond to color. They like red substrates. They respond to different types of chemical cues. So there's an amazing hierarchy and sophistication that those little teeny blobs that have no brain and no neurons are able to use to choose a place where they settle. And so I think it's logical to expect that as we change the ocean environment with a lot of different types of chemicals and maybe changing the turbidity or the light regime in the ocean a lot of times, we're certainly changing the sounds that they hear, right, with lots of boat traffic and different types of things. I think it's plausible that just the cues or the roadmap that coral larvae are used to using to find their home is just all messed up. Again, that's not something that's easy to prove, but I think it's logical that a lot of the changes that we're making in the ocean make it harder for them to find their home. And then after the ones that do settle, as I mentioned, that picture that I showed you, there's various aspects of reef communities that have changed over the years. And a lot of that has to do with herbivory and grazing. And that's really the most important factor, as best we understand, in terms of enabling the corals after they settle to be able to grow up and keep that algae and those other competitors kind of at bay. So there's both grazing fishes that are very important, but also, how many of you remember the big black spiny sea urchins? There aren't too many around these days. They're very rare now. But back in the 80s, if you went snorkeling back in the 80s, anywhere in the Caribbean pretty much, they were a menace. They were a hazard, actually. Those black spiny sea urchins are called Diadema antillarum. And they all died in around 1983, 84.
There's plenty here, though, in Key Biscayne.
A couple places. They're real patchy. There's a couple places where there's...
You know the hard coral, they're always... There's a
nestled right underneath. Yeah, and a diadema. So there's a few places they've come back, but I said they all died pretty much in 83, 84, and they haven't really come back, consistently across the region. Now those diadema, those urchins, they're like little lawnmowers. They just go along and like mow down the whole surface of the reef and keep it clean. And we have a pretty good understanding that keeping that surface clean, that's the habitat quality that those baby corals need to survive after they settle, or maybe to enable them to settle as well. So I'd say there's three phases of that are all a little bit or a lot messed up that make it really hard for baby corals to make it on their own. So those, again, are all things that we need to be working on fixing. Yes?
Hi, the ingredient in the sunscreen that you mentioned, why is that bad for?
It's called oxybenzone. So there's pretty good scientific, I sort of alluded to, it's very difficult in the sort of the field of toxicology because there's so many different chemicals that go into the ocean. And being able to pinpoint individual ones that are causing harm is very difficult scientifically. But in the case of oxybenzone, which is a very common ingredient in the sunscreens that we use, there's very good laboratory experiments now that have been done that show both that it kills larvae and that it impairs that photosynthesis, again, the symbiosis that's really important to the adult corals. It also creates a lot of impairment for that. So they're very direct toxicological effects of that chemical on corals, both adult corals and larval corals, at levels that can be measured at very popular swimming beaches. So again, the authors of this paper sampled water like from really popular snorkeling beaches like in St. John and in Hawaii. And they could measure, I would suspect so. There's probably similar beaches and similar areas with very high visitation in our region, but they were able to measure levels in the water, in the ocean water, that were at a level that caused harm in these laboratory experiments. And so this is really the basis for sort of singling that one item out and saying it's good to avoid, right? Not that it's going to solve all the problems for corals, but it's something that's easy to change and we know is causing harm when there's a lot of it going on. I want to emphasize too, though, so that's mainly from people that are in the water, right, where you have a beach or a snorkeling area or a popular reef where there's a lot of people in the water that, you know, slather the stuff, pour it in the water, whatever. But I want you to think about, too, anytime you put that on your skin, the end of the day or the next morning, you're going in the shower, right, and you're washing your skin off. And so any of that stuff that we put on our skin every day is going down your drain eventually at some level. So that's the other thing to remember. Anything that you use in the way of personal care products, any drugs that we take through our bodies, where do they come out? A little bit of it gets metabolized, but where does the rest of it go? It goes down the sewer. And where does our sewer go? It goes into the ocean eventually. You know, there's various types of treatment. But honestly, any of our sewage treatment, it does nothing for any of those pharmaceutical chemicals that are going through our sewage stream. So, you know, just be aware of that. Be aware of anything you put on or in your body. As I said, on your car, on your lawn, you know, anything that interacts with the rain or our water system, it all ends up in the same place. So those are all factors that are important, as I said, in terms of affecting the environment where corals are trying to live.
I think actually with the oxybenzone too, they found that it reduced the temperature that corals bleach at by like roughly a couple of degrees.
It's significant, yeah. So again, so that means it's making them more sensitive to that heat stress that causes bleaching. So when you do have heat stress, it's just having that chemical pollutant as well makes them more sensitive. Exactly. Those are great questions. Yes.
Well, I would say more than a question, it's a comment.
Sure.
I go out to the reefs out here. We have a huge area in Key Biscayne of reefs.
Yes.
Most people don't even know about.
Absolutely do.
And I mean, I go out again every single week that I can on my kayak. I just go out there to the reefs.
Excellent.
And it's mostly soft corals.
Yes.
We have some hard coral too, but I would say comment is that I've never seen any coral being propagated here. Nowhere. And I dove in maybe, I don't know, 300-400 times. So
there is a fairly active coral propagation effort based on fragments. There's a group at Rasmus at the University of Miami, and as I said, they have a citizen science component called Rescue Reef. So they are growing mostly staghorn coral, and they are outplanting it throughout the county. Now again, there is a huge impact from the hurricanes last year, and so they had a huge setback from that. The physical disturbance stuff has caused problems, but it's when they go to outplant the corals, so when they take the corals to actually go and use them on a reef, there's a lot of factors in terms of choosing, you know, a site where they're going to hopefully be successful. So it may be that even if you've been a lot of places, you haven't managed to be to one of the 20 sites or something where they've actually done outplanting.
I would love to do something like that.
Yeah.
Help with that.
So yeah.
If I can add to that.
Please.
So we work with Rescue Reef.
Right, that's my thought.
If you want to put your name on our mailing list.
Can get you hooked up with them.
We had a trip planned with them last September, and it was a week before the hurricane. And so we canceled it because the hurricane was coming, and their nursery got really heavily damaged.
They had a lot of damage.
We are planning on hopefully making it an annual trip, so we are still hoping to schedule another one this year. And yeah, so I mean, if you're on our mailing list, I'll be sending out notifications of it once we do it. And we have a site actually just off of Key Biscayne where we do our planting every year.
Okay.
There you go. So that's a great effort to get hooked up with.
Definitely. And I saw also a big change in the seagrass after the hurricane. There was almost no seagrass.
Hurricanes are damaging.
It's important for the little fish.
Yeah. Hurricanes are tough. I'll just mention too. So Secore, we don't currently have active projects going on in South Florida. As I said, our primary sort of testing grounds are in Curacao, which is in the Southern Caribbean, and the Yucatan Peninsula in Mexico. And now we're just ramping up our operations in the Bahamas, actually, so close across the way. But we're anxious to begin doing some pilots here in South Florida as well. So hopefully in the next year or two, we'll also be raising baby corals here, but that isn't, we're not actively doing restoration here in South Florida yet, but hopefully soon. So stay tuned on that. Any other questions or comments? Yeah?
Do you engage in any other projects other than the coral, the Secore?
So the other piece that we've talked about some, and we haven't taken action, we've talked about a lot actually, is those Diadema, those urchins that I was talking about. Because that is another very functional piece of Caribbean coral reefs that's kind of absent in most places still. And as I mentioned, they haven't recovered. They probably have some of the same issues of larval limitation. Again, when there are not enough parents, there's probably not enough larvae being produced in the first place. They have a really long larval period too. They have a complicated life cycle like that where they have little tiny babies that float around for like a month, rather than like the coral babies only float around for a week. So it's an even larger challenge to sort of culture and propagate them. But there's been a lot of research done on that as well. And we have an idea that some of our little substrates maybe could be also nice little habitat substitutes for baby urchins as well. So that's something that we've been thinking about getting involved in here in Florida, but it's still a little bit in the idea stage. So, but again, a really important organism to the reef that's really mostly functionally lacking.
The grass that it competes with
The algae.
The algae that the coral. Do they have a specific beneficial function for the life of the coral as far as?
So the ones that compete, the ones that grow on the reef bottom, they don't necessarily benefit the coral. They do, they do plant function for the reef system. So it's kind of like the grass in a reef. So they photosynthesize, they provide food for a lot of the herbivores on the reef, this sort of thing they provide. So they definitely have their ecological function, but the trouble is they can grow a lot faster than corals grow. The corals grow relatively slowly. And so it's easy for them to kind of overtake the corals when they're really small. But no, they're a normal part of the reef. They belong there. It's just hard to keep them in balance, as I said, without as much of the grazing fishes or those urchins around to kind of help keep that balance with the slow-growing corals.
Is the urchin the only animal that will?
It's the most effective one, I would say. They're amazing how much those guys can mow down. But like parrotfishes, for example, also serve a similar function in terms of being able to eat the algae down and scrape the reef surface clean. Surgeon fishes as well, like tangs. I don't know if you how many know your reef fishes, like blue tangs and doctorfish and things like that. Those types of fishes are also herbivores, and so they will eat the same, those algae as well.
Do hogfish also do the same thing? Because they're always on the bottom.
They pick around. They're more picking in the sand for little invertebrates. They're not herbivores. They're looking for little like worms and stuff like that are down in the
bottom or no?
Yeah, so they're picking around at the bottom, but they're looking for animals. They're not grazing on the plants. So.
And you take civilians to help out on these?
So it's a really good question. For the work that Secore does, or at least the way that we're doing it now, it's difficult to incorporate citizen science because a lot of the diving is very challenging. As I said, all of our diving work goes on at night, pretty much. We're hauling these big loads of heavy substrates around and doing kind of crazy stuff. So it's something that we're thinking about. We're thinking about a lot in terms of ways that we could help people be involved. But we haven't quite figured it out very well yet. But as I said, there are definitely other opportunities to be directly involved through some of the local efforts, and those are certainly available to everyone. So. Thank you, everyone, for your interest.